right. Well, hello, everybody. Happy Valentine's Day. I'm Beth Johnston. I'm the Chief Community Engagement Officer here at the FSHD Society. And together here with my colleague, Ms. Jun Kinashida, our Director of Research and Patient Education, we'd like to welcome you to the very first FSHD University webinar of 2023. For those of you who don't know us, the FSHD Society is the world's largest research-focused patient advocacy organization that is focused solely on FSHD. We've got over 30 volunteer-led chapters across the US and growing in Canada now, and we're part of the 20-plus country World FSHD Alliance. And our mission is to find treatments and a cure for FSHD while, of course, empowering you, our families. And we offer these free FSHD University webinars and so many other wonderful educational events to educate and empower everyone that's affected by FSHD to ensure that they are their own best advocate. So today from Avidity Biosciences, we welcome Amy Halseth and Alyssa Peters. Amy is the Executive Director of Clinical Development there, and Alyssa is the Director of Patient Advocacy and Corporate Affairs. They are going to speak to us today about FORTITUDE, which is their phase 1 and 2 clinical trial in FSHD of their compound called AOC 1020. I think that's right. So welcome, ladies. I will turn the platform over to you.
Great, thank you. Perfect, so you should see that okay. So I just want to start by thanking the FSHD Society for inviting our team to share about Avidity and our progress in developing potential treatments for the FSHD community with you all today. Today, we're really excited to share about our recently announced study, FORTITUDE, which is the phase 1/2 clinical trial investigating the potential for AOC 1020 to treat adults with FSHD. This slide has a lot of text, but it's really just to say that Avidity is a publicly traded company, and we will be making forward-looking statements during today's presentation. For today's agenda, I'll start by sharing an overview about Avidity Biosciences and who we are, our technology, and our approach to patient-centric drug development. And then my colleague, Amy Halseth, Executive Director of Clinical Development and Avidity's FSHD Program Lead, will discuss our development program, AOC 1020, and share about the FORTITUDE clinical trial. And following that, we'll be happy to answer any questions that you have. So I'll start by talking about how we approach patient-centric drug development here at Avidity. Avidity is a biotechnology company based in San Diego, California. At Avidity, patients are at the heart of everything we do. Our mission is to improve the lives of people affected by diseases who have limited therapeutic options, including FSHD. And we're really aiming to do this by advancing a new class of potential treatments. Our team's extensive experience in drug development, combined with our commitment to involving patients and their families at every step of the way, is truly the foundation of our company and is what drives us in advancing new treatments for rare muscle diseases, including FSHD. Our innovative drug technology unlocks the new way to deliver drugs directly to affected cells like muscle cells. And this allows us to develop new treatments for diseases where there are limited therapeutic options available today. We're applying this technology to develop a new investigational treatment for FSHD, AOC 1020, which has the potential to address the underlying cause of the disease or the root cause of the disease. We strive to continue to partner with the FSHD community to incorporate the patient voice into everything we do. And for the focus of today's presentation, we're currently initiating the FORTITUDE study with AOC 1020, which you'll hear more about from Amy shortly. So first, I'll talk a little bit about our technology. At Avidity, our potential treatments are called antibody oligonucleotide conjugates, which is a mouthful, so we say AOCs for short. We have specifically designed our lead AOCs to target the root cause of disease in muscle. And we're developing AOCs with a goal to improve the lives of people impacted by diseases who have limited therapeutic options, including FSHD. The AOC platform combines the proven technology of existing drug types that are really well understood for their specific properties in drug development, antibodies and oligonucleotides, or oligos for short. An antibody is a naturally occurring protein made by your body to help fight infection. Antibodies are commonly used in drug development because they can be made to target almost any protein, including those that are different between one cell type and another. Antibodies are well characterized for safety and are actually the foundation of over 100 approved drugs, many of which are used for chronic treatment. At Avidity, we're using an antibody as a vehicle to deliver drugs directly to muscle cells. And oligos are short strands of DNA or RNA, and oligos can be very powerful because they can change gene expression or how the body makes a protein by targeting RNA. There are currently multiple approved oligo drugs, many of which are disease-modifying treatment. And at Avidity, we're using oligos in our AOCs that are created specifically to target the root cause of disease. So for muscle diseases, the antibody component of the AOC enables targeted delivery to muscle cells, and this in turn improves the power of the oligo. The antibody has a unique ability to recognize and stick to a specific protein on muscle cells, delivering more oligo to change gene expression. For our AOCs in development for FSHD, the antibody binds to a transporter protein called the transferrin receptor 1, or TFR1, which you can see here in the diagram. And this is in the muscle cell to allow the oligo to modify gene expression by targeting RNA. You can think of the antibody as a bus that's taking its passengers, which are the oligos in this case, to a very specific bus stop, and then letting off its passengers to go do their job. In this case, the bus stop is the muscle cell, and the oligos are able to enter the cell and do their job more efficiently. By combining these two drug components of the antibody and oligo, AOCs have the power of the oligos to modify gene expression and the ability to target specific cells in the body using the antibody. This allows us to develop new treatments for diseases where there are limited therapeutic options. Here you can see our research and development pipeline. We are applying our AOC technology to develop a pipeline of potential treatments for rare muscle diseases, including FSHD. At Avidity, we seek input and partnership with multiple key stakeholders in the development of new treatments. We believe that it's only through the active and ongoing partnerships across stakeholders, including patient advocacy organizations like the FSHD Society and the community of families they support, healthcare providers, health authorities like the FDA, and coverage and insurance providers, that true patient-centric drug development is achieved. These collaborations can help ensure that patients and their families are put first in decisions in our shared effort of advancing new meaningful treatment options. At Avidity, we're continuously seeking input from the FSHD community throughout each stage of the drug development process to best address your needs. Collaborating early and often will always be a part of our process, and we are doing this through listening, partnering, and connecting with families in the community. Patient and caregiver perspectives inform our approach to drug development and inspire us to stay committed to our mission to improve the lives of people impacted by conditions, including FSHD. And finally, we want you to feel heard and supported and know that you always have an open line of communication to our team. And we'll share an email address where you can reach us directly at the end of this presentation. And on this slide, I want to share some important examples of how your voice has already directly impacted our program for FSHD AOC 1020. Through partnerships with organizations like the FSHD Society, we're grateful to connect with you in forums like today's webinar, as well as support the important resources and initiatives that these groups bring to the community. We are also investing in gaining a deep understanding of the journey of living with or caring for someone with FSHD. Through conversations with you, we have gained important insights into the experiences and challenges in your personal journeys to a diagnosis and establishing your care team and your management plan. We have also brought together a focus group of caregivers and patients from the FSHD community to inform the design and services of the FORTITUDE trial, which you'll hear about shortly. And finally, we're collaborating with community leaders in an ongoing way with the goal of addressing gaps in support services and resources available. So thank you to all of you from the community for partnering with us as we advance toward our shared goal of bringing new therapies to the FSHD community. And now I'd like to turn it over to my colleague, Amy Halseth, to talk about the FORTITUDE study.
All right, thank you so much, Alyssa, and thanks to everybody who's logged on and spending some time today listening to us talk about AOC 1020 and the FORTITUDE trial. I think June said there were over 500 people who signed on to listen, and that's super touching to us that how invested you all are, and really speaks to how special this community is. So Alyssa, if you can go ahead and advance the slides. So it's great because Alyssa set this up so well, so I can talk a little bit more about specifically how AOC 1020 works, which is the compound that we'll be studying in the FORTITUDE trial. So as she mentioned, all of our drugs are AOCs, so antibody oligo conjugates. And so I'll start at the beginning again, just to go through this. So we have the antibody linked to our oligo that binds to a receptor on the membrane of muscle cells, and that allows the oligo to be brought inside of the muscle cell. That compound or complex dissociates once it's inside the cell, and that frees up the oligo to really be able to go to work, as Alyssa said. And what it does is it basically hangs out in the cell and looks for any DUX4 messenger RNA that's being made, and it's able to bind to that messenger RNA and stop DUX4 protein from being made. So as when Alyssa said, we're going after the root cause of FSHD, we really are. And by doing that, we're trying to just block any of that inappropriate DUX4 gene expression. Then you don't get that DUX4 protein, and then you don't get some of those toxic downstream effects that can lead to the symptoms and long-term consequences of FSHD. So if you go to the next slide. So how do we know that this works? The oligo that we use is something called an siRNA, which is a small interfering RNA. And that has a sequence that's complementary to the mRNA of DUX4. So it binds to it, and then you have endonucleases within the cell that will destroy it. And what that leads to is an impact on the downstream consequences, like I said, of DUX4. So I'm going to walk you through the slide, and there's a lot on here, but it's actually pretty simple when you go step by step. So we'll go ahead and do that. So one thing is it's really hard to measure DUX4 expression itself. So what we measure are genes that are downstream of DUX4. So we know that when DUX4 gets made, it turns on a bunch of other genes that probably shouldn't be expressed in skeletal muscle. And so, but we can measure those and use those as a tool to understand what's happening within the cells. Okay, so in this experiment, we have some what we call myocytes, which are derived from skeletal muscles taken from biopsy from patients who have FSHD. And you can see that we then can increase the amount of our oligo, increase the amount of our siRNA. And what we do is we can decrease the amount of that DUX4 regulated gene expression. So as you move to the right in the figure, you increase the concentration of oligo that you put on those cells, you decrease the amount of that DUX4 regulated gene expression. More than an 80% reduction in the expression of those bad downstream genes. So if you go to the next slide. So that was just some muscle cells in a dish where we did that last experiment. We've also taken our compound into animal models, and we've been able to show the same thing with significant repression of the DUX4 regulated genes. So again, there's a lot on here, but we can walk through this together and go through what's shown here. So this is an experiment in which the antibody linked to our siRNA is given intravenously to a mouse, and then we're able to measure that DUX4 regulated gene expression in the mouse skeletal muscle. And so if you start just on the day, I love all the chats, but they're popping up right in front of my slide here. Okay, so day 14 is the first time we look. And if you just start looking at sort of that green dot and then the two blue, that's just when we give that's 14 days after we've given our AOC drug to these mice. And you can see that as you move from a lower dose to a higher dose, you get additional knockdown, going from about a 20% knockdown at this low dose to about a 75% knockdown as you move to those higher doses. And that's at that first time point we look 14 days after. So then the other part of that experiment is if you now look out across time, what you can see is that as you go from day 14 to 28 to 42 to 56, you still see about that same magnitude of reduction of those DUX4 regulated genes. So we know from this experiment that a single IV injection into a mouse, anyway, can give you that same sustained suppression of DUX4 for at least 8 weeks. We've done a lot of different experiments in mice and other animal models that we believe that this is something that will be consistent in humans as well. So this will be an intravenous infusion, but we believe we can have a relatively long dosing interval between when people will get their doses of drug. All right, so if you go to the next slide. And so taking it one step further, that was just looking at the downstream genes of DUX4, right? So we saw an 80%-ish reduction in the DUX4 regulated genes. And I did see a question in the chat. We can't really measure DUX4 specifically just because DUX4 is expressed at such low levels. It's just very difficult to measure itself. So we use those downstream gene markers as kind of an index of how much DUX4 we think we're inhibiting. But now coming back to this experiment, this is a different mouse model that will actually develop FSHD-like symptoms when you give the mouse tamoxifen. So when you give the mice tamoxifen, it turns on DUX4 in their skeletal muscle, and they'll develop symptoms that are like FSHD in a pretty short time frame, about 2 weeks. So actually, if you look on the bottom left where it says recovered running, you can see, so control is those mice who haven't been given tamoxifen. If you give them tamoxifen and you turn on DUX4, and then 2 weeks later, look how well they're able to run, you can see they can hardly run. In this specific experiment, they go from being able to run 500 meters, which is actually the max in the experiment. They can barely run 100 meters just from having that DUX4 gene turned on for 2 weeks. And similarly, if you measure the force in a very specific assay that measures muscular force, you can see that in that blue line, from giving that tamoxifen, turning on DUX4, those muscles are very weak and aren't able to generate the same amount of force. Now, in our experiment here, what we did is we gave an AOC drug 2 weeks before we give the tamoxifen. And what that does is it blocks okay, so we give our drug. Our drug is sitting there in the muscle. When the tamoxifen is induced and the DUX4 is supposed to be turned on, our drug is sitting there basically waiting, looking for any DUX4 messenger RNA that's being made and destroying it. And so what you can see when you then look at the phenotype of these mice and look at how they're behaving, they're able to run just as well as the mice who didn't get tamoxifen. You can see on the far left here with running. And similarly, when you look at the force, you can see that they restore the amount of muscle strength that we would have seen in the absence of any DUX4 being turned on. So it's really nice to see this proof of concept in a mouse model that, yes, this drug is getting into muscle. It's able to bind the DUX4 mRNA, and it's able to block the downstream negative effects of having that DUX4 mRNA turned on. So you can go to the next slide. So that's great for mice, right? And then there's a lot of things and a lot of diseases, a lot of conditions where we're able to cure things in mice. The real question is, okay, well, what happens in people? Is this drug going to have an impact on FSHD in people? So if you move to the next slide, this is what we're going to be studying in the FORTITUDE trial. So the FORTITUDE trial is a phase 1-2 study. And so what that means, so phase 1 is always the first stage of drug development where you're really trying to look at safety and tolerability. It's a very important question. Obviously, for any drug that starts, that's the foundation. It needs to be safe. But because we're calling this a phase 1-2 study, what we're also able to do in this study, both look at the safety and tolerability, as well as try and get some early indications of what we think the drug could be doing in terms of efficacy and in terms of some of the symptoms of FSHD. And so here's the overview of the study. We can go through that. So it's a phase 1-2 study, as I mentioned, of AOC 1020. And this will be the first time that AOC 1020 does go into humans. And we plan to enroll 72 people who are ages 18 through 65. And people are going to be randomly assigned to receive multiple doses of either AOC 1020 or a placebo via an IV infusion. And it's really important that we have a placebo in this study for a couple of reasons. We want to make sure that we understand the safety of the drug and not just the safety of having FSHD or side effects or things that would just happen in a person with FSHD. We really need a placebo group so we can understand safety-wise, is there a difference if you get the drug or if you're just living with FSHD? And similarly, as we move into some of these exploratory markers of trying to understand the efficacy, we know that FSHD is really variable and it's also very slowly progressing. So what we want to try and understand is the drug doing something different than just, the normal course of living with FSHD. Part of understanding how the drug works is we're going to be taking muscle biopsies as well as MRIs to be looking again at those DUX4 regulated genes and trying to see if we are having an impact on DUX4 regulated gene expression, and also looking at using MRI to get a sense of muscle volume and muscle fat. So some really important biomarkers of FSHD activity. And this study will go out to 12 months in terms of the follow-up. And we're also planning to have what's called an OLE, or an open label extension study after that. And in that study, whether you've received placebo or AOC 1020 during the original study, everybody would receive AOC 1020 going forward. And that allows us to get longer term safety data, as well as more understanding of the potential efficacy of this drug. So as I mentioned, the primary goal really to understand safety and tolerability. But then in terms of some of those other effects we're looking at, it's things like muscle strength, what we'll be asking, having you fill out, or the patient population, a lot of questionnaires about their FSHD and about their symptoms, as well as quality of life. So really trying to get a lot of information from this one study. Next slide, please. So this is what the study design looks like when we put it into a figure. And again, there's a lot on here, so let me walk you through it. We have the study basically broken down into 3 parts, which we call Part A, Part B, and Part C. And so in Part A, again, this is the first time this drug is going to go into patients. And so we start with low doses, which is really how most drugs are studied. We want to start with very low doses to make sure it's safe. And we want to start with a relatively small number of people as we go forward. And then we would then take a look at that safety data with an outside group of doctors who are experts in FSHD and statistics and clinical trials. They help us review that data, and then they would give us the go-ahead, hopefully, to move into Part B, where we can study higher doses. And then based on that, we would then also move into Part C. And there we would want to get more data on the dose or doses that we think have the most total potential. So that would be Part C, and at a dose to be determined. So, but for all of these parts, whether it's A, B, or C, they have a 12-month follow-up period. And over that time, people would get 5 doses of study medication through that IV infusion. So they're generally given quarterly, except you can see that little IV bag icon thing with the dots around it. That would be a booster dose. So the way that everybody will be dosed would be like basically on day 1, week 6, week 13, and then quarterly thereafter. And we believe that roughly quarterly is probably going to be the appropriate interval for dosing based on those animal studies that I showed earlier and what we know about how siRNA, what the stability of siRNA in skeletal muscle is. And you can see the numbers go up as we move forward, where we start with a relatively small group for Part A, and then as we move into B and C, we have additional patients there. I think we can go to the next slide. Yeah, so who are we studying? We already mentioned it's for 72 adults. We're starting with adults for now. We do have a lot of interest in going to younger groups, but right now the way trials are typically done is to start with adults. And so for us, it's males or females aged 18 to 65 years old. For this first study, because of some of the functional tests we have, we need people who are able to walk 10 meters with or without assistive devices. So 1 cane would be okay. If you need 2 canes or a walker to cover 10 meters, you would not be eligible for this first trial of AOC 1020. And again, that's because we need people who are able to do the tests so we can look for either improvements or worsening over time. So you need to be able to do those tests. And again, for this study, you can't have been diagnosed with congenital or infantile FSHD. We're looking for sort of the more traditional, I'm not sure what the opposite is, adult onset FSHD. And you can't be pregnant, breastfeeding, or planning a pregnancy in the next 1 to 2 years. Okay, so where are we doing this? I was seeing all of the locations come in during the chat when we were waiting to get going, and I was getting excited because a lot of people live close to where some of these sites are going to be. So we're in the process right now of initiating our sites and getting them activated. So we don't actually have site names yet, and we aren't ready to have people start calling sites to try and get into trials. But you can see that we have a nice spread here from the West Coast through the Midwest to the and running in the next 1 to few months. And there is a second wave that will be planned of site activations that are planned to happen in Europe as well. So I did see a lot of people in Europe in the chat. I would say just keep paying attention. And in particular, by the time we get to sort of those B and C cohorts, we think that there would likely be some sites activated in Europe and the UK as well. So in terms of knowing where those sites are and how do you stay involved, if you are interested in potentially participating or have a family member who would be, we will be having a listing on clinicaltrials.gov. And the way that works is as more sites become activated, more sites get listed on that website. And then also through the FSHD Society, we'll be partnering with them to help share the news about when sites are active and how things are happening there. So there will be more information coming very soon. But just know we're working as hard as we can right now to try and get all these sites going, because we know that there is a lot of interest in people in trying to get into these studies. Okay, next slide. So it's exciting, and it's really a huge opportunity to be able to put this trial on and to potentially be a participant in a clinical trial. You know, I think we have a lot of optimism and a lot of belief that our medication could potentially have nice positive impacts, but it is also a lot to be in a clinical trial. If you haven't been a participant in a clinical trial before, there are a lot of visits, there are a lot of measures, there's, the muscle biopsies, there's a lot of, blood draws and questionnaires that you're asked to figure out. So it is, it's not an easy thing to do. And I think deciding to join a clinical trial is a decision that should be taken seriously. And we want people who would be involved in this to really understand what they're getting into. And especially given that this is the first time this has gone into humans, there are even more visits than you would typically have like in a later stage trial, just because there's a lot we want to pay attention to make sure that everybody is safe and well taken care of. That said, we have tried as much as we can within that framework to take the patient voice into account and try to put some things into the trial that could make it somewhat easier for somebody to participate. And so some of those aspects are here. So one is home health. So whether you live pretty close to a site or you would maybe have to travel, sometimes just even going into a site can be quite a bit of an effort, depending on the parking. And, even for me, like going to my doctor's office, like the parking structure is like 2 blocks away from where my doctor's office is. And, these big hospitals, sometimes that can be a challenge. So we have about 1/3 of the visits that are planned for the trial. Instead of you having to go anywhere, it's the potential to have a travel nurse basically come to your home and draw blood or take your vital signs and do that kind of thing to try and lessen some of the burden on the participants in the trial. We know that there is not a site in every country or even every state or every city. So another thing that we have built into this trial is what we call a travel concierge. So if you are an excellent participant and you have the ability to handle all of that, because we know that we want to get the best potential participants into the trial. And so if that means we have to have some people travel to sites, that's something we can help with. And there are some expenses that come with being in a trial, whether it's the travel, as we just mentioned, or even if it's a site local to you, meals, parking, if you're there over a couple of days, maybe you live 2 hours away, you don't need travel assistance necessarily, but if there are visits that occur over a couple of days, maybe it's helping with a hotel, there's a lot of reimbursement that Avidity will do to make this possible, because we don't want people giving so much of their time and effort and also having to pay for it financially. So we want to try and give that support as well to the patients. Okay, I think next slide. Yeah, so just kind of to wrap up here, and then I know I'll have some time for questions. Just to remind you, so AOC 1020 is being studied, and what's really exciting about this is it should treat the root cause of FSHD in muscle by going after DUX4 specifically. We really hope it can slow the progression of FSHD. And what we don't know, the real question is, could it even potentially lead to some reversal of some symptoms? This is only the first trial. We won't be able to answer all of the questions now, but that is really the big question that we hope to answer over the next couple of years. AOC 1020 would be administered by infrequent IV dosing, roughly every 3 months, specifically in this phase 1/2 FORTITUDE study, which we'll be initiating very, very soon. We've done our best to design this trial in collaboration with patients as well as advocates and key physicians in the field. So hopefully by the time this all comes together, we really want to have a good understanding of the safety and potential efficacy of the drug. So hopefully while there's a lot in this study, it will allow us to answer a lot of questions. And in collaboration with the FSHD Society and other patient advocacy groups, we'll be working really hard to get you more information and resources as soon as they're available. So I think I have one more slide here. So this is maybe 6 months ago. So I think our team is actually even bigger now, but this is the Avidity employee group after a on-site team building / work week thing that we had. And, there really are there are real people at Avidity who are working to try and bring this drug forward. And our CEO says, we're people working together to help other people. And it's just really permeates everything we do. I think Alyssa described really well how patients truly are at the center of everything we do. And it really is what motivates this team of very happy, dressed in gray people here. It really is the ability to do this work and potentially bring new treatments out to the patient community that motivates us. And as Alyssa promised earlier, also, here's the email address, patients@aviditybio.com. If you want to reach out, that'll come to us. I'll be able to answer additional questions, keep you on maybe well, I don't want to promise what Alyssa actually does once you email her, but there's the email address. So I think with that, we will turn it over to the Q&A session. Is that right?
Perfect. Thank you. If you can unshare your slides, we can all see you on screen. And you can tell the audience is very excited. They haven't been able to stop chatting yet. So with that, yes, why don't we jump right in? Here's a question. Are there any approved therapies for other diseases using this kind of AOC approach? And if so, how effective are they?
Yeah, so right now we are the first to develop this AOC approach. And so far there are no other approved therapies, but it is, as Alyssa mentioned, a mix of two things that do have a lot of drugs. So monoclonal antibodies are used for a lot of conditions like rheumatoid arthritis and many other, a lot of immune diseases use monoclonal antibodies. We're using them differently, right? We're just using them as the bus. But monoclonal antibodies have been used a lot. And then oligos are used, but they've historically really only been able to be used mostly for like liver, because that's where oligos go if they don't have an antibody with them to help bus them around. So what's unique about us is this is the first time we're able to really harness the power and the selectivity of the oligo and get it into skeletal muscle where you need it for FSHD and other rare muscle diseases.
Great, thank you. Here's a question about what happens to the antibodies after they drop off the oligo. Does the body attack the antibody? And also, does every muscle cell receive the oligo?
Yeah, so Oh, those are good questions. So what happens is the whole complex of the antibody and the oligo get taken into the cell together. They then go through some compartments where things sort of get split apart and then chewed up. And then what happens is the antibody is thought to be just kind of cleaved in the cell through some lysosomal degradation. So it doesn't get kicked back out and circulate around. It basically comes in and then it gets degraded within the cell to sort of its little components. And then those are just basically turn into like amino acids in the body. And then, sorry, there was two questions and I forgot the second one.
Oh, geez, let's see. I just, hold on. Let me go find
It was, does the oligo go to every cell,
every cell in the
muscle?
muscle.
Yeah.
Thanks, Beth.
You know, we think so. You know, it's a hard thing to measure specifically. The fact that we're getting such good knockdown makes us believe that it is very widely distributed within the muscle. You know, muscle is really highly vascularized. There's lots of capillaries around each of the cells. So you have a really good perfusion of that system going through the muscle. I can't promise it goes into every cell, but I think the fact, you know, regulated genes would lead us to believe that most muscle cells are going to have some of that oligo in there.
There's a question asking you to comment on the critical difference between something like this, the FSHD approach, where you're trying to suppress a toxic gene, versus something like a treatment for Duchenne, where you're trying to replace a defective protein. What are the comparison, I guess, in terms of chances of success as a, you know, therapeutically?
Yeah, so there's a lot there. So, you know, in Duchenne's, where you essentially have no dystrophin, we know that it doesn't take very much of an increase in dystrophin to have really profound impact on the muscle. What our program is doing specifically in Duchenne's, and what a lot of them are doing, what happens in dystrophin is it's a very big gene. And what happens is that you get kind of off in terms of the reading frame, and it becomes like a lot of nonsense. You can't make the protein. We're using a different kind of oligo in Duchenne's that causes a differential kind of splicing and basically puts you back in the correct reading frame. So you don't make sort of ideal dystrophin protein that's better than no dystrophin protein, and you don't need very much to have a big impact. That is very different. And I think in some ways, our approach is easier. Like, we know who the bad actor is in FSHD, right? It's DUX4. And that's what siRNAs do really, really well. They find the bad guy, they hybridize to it, and then they can help get it chewed up. So, you know, it'll be interesting to have both of these programs going forward at the same time, and we hope they'll both be successful. And they both have their own unique challenges, but we're hopeful that we can have efficacy with both.
You're undaunted. You're going to do it.
We have fortitude.
Yes. Question about what type of biopsy will be conducted. Is it a needle or an open biopsy?
Yeah, we'll be doing two needle biopsies, one at baseline and one after people have been receiving the AOC 1020 for a while. And that's really to look at two things. One is to look at the DUX4 regulated gene expression, and one is to look at the muscle concentrations of the AOC 1020. So like the amount of drug that's in the muscle.
This is a question about using the downstream genes as a stand-in for the presence of DUX4. And why is that done? How solid is that?
Yeah, so DUX4, you know, it's a squirrely little guy, right? So for all the harm it does, it's really thought to be expressed in about 1 in 1,000 nuclei at a time. So it's really tricky to measure with our technologies. And you can imagine that if you have just a biopsy in a pretty small area, and then a biopsy somewhere else a little later, even if you happen to get lucky and kind of get a little chunk of tissue that does have some DUX4 in it, it's going to be really, really low. And you might take another sample another time. And even if you got lucky and were able to like barely detect it in one, you just probably might it would be unlikely to be able to detect it again. So really the whole field has focused more on looking at downstream targets of DUX4. So DUX4 itself is what they call a transcription factor. So once it's made, it turns on a bunch of other things that aren't typically expressed in muscle or expressed at very low muscle. So it really is how most drug companies and even academics really try and get a sense of DUX4 activity is from looking at these downstream genes. That said, they're still super variable, because if you think of that 1 in 1,000 thing, you could imagine kind of one little area might have a lot of DUX4 and then a lot of the downstream genes, and then you move over 3 inches or something within the same muscle, you just might not even get the same expression of all of those other genes. So even using the downstream genes, which are easier to detect, there's still a lot of variability. And we know that it's not a slam dunk that we'll even be able to really show anything in terms of the DUX4 gene expression, just because it's, going up and down like that. So that's why we really hope we can see it, and we would love to be able to see it. That would be a great proof of principle for us for this whole program. But that's part of the reason we're treating patients for out to a year, because we also want to be able to look at some of these functional effects, your strength and mobility measures, so that if we don't get kind of lucky on some of the DUX4 gene expression measures, we have other things to look at to try and get a sense of whether the drug is having an effect on muscle.
Okay, you probably mentioned this and I don't recall, but is the biomarker, is that your primary endpoint and the functional markers are your secondary endpoints?
No, the primary is actually safety and tolerability. So given this is a phase 1 study, and we're, there's not a lot of data about, how big of changes we would expect to see if we are suppressing DUX4. So, when you do this first study, you usually have relatively small sample groups. You're looking for trends. If we would move forward like into phase 3, then you would have an efficacy endpoint would be usually the primary. But in this stage, you almost always have safety and tolerability as primary endpoint.
So this would be something you need to extrapolate, I think, from the animal data. But when you see a mouse that you express DUX4 and it's got various impairments happening, does the AOC simply stop the progression, or does it actually provide for the ability to recover for the muscle recover function?
So it's interesting because that mouse model has some quirks. It's great for the very specific way we used it of being able to demonstrate, like, yes, you can quickly sort of induce a FSHD like phenotype, but like I said, it's 2 weeks. So think of, that's not how FSHD works in people, right? That is like a massive, like DUX4 is boom on, and we can block it. What you can't do very well in that mouse, what's hard to do in that mouse model is have a low level of DUX4 consistently, and then treat a mouse like you would treat a human, where they already have mouse FSHD, and then you come in with our drug and see if you see improvement or at least a slowing. It's just a hard experiment to do, and we are exploring ways to try and get at that. We just haven't been able to do it just because of the way the mouse model, that specific mouse model works.
Okay. There's a question here. What is the difference between an RNA-based and a therapy and a gene therapy? I think people hear RNA, DNA, like, what's the difference?
Yeah, so I think, well, you guys probably know more than me about gene therapy, but with a gene therapy, I think what you're trying to do is go in and change the DNA in a way, so that you would like maybe change the chromatin structure so you wouldn't even get DUX4 expressed, with like other ways of addressing maybe even a little bit upstream of where we're working. With an RNA therapy, we're not changing the DNA within the cells. We're just sitting there waiting, and if a DUX4 molecule gets made, we're hopefully going to bind to it and destroy it. So we're just a little bit further downstream in the process of how you go from DNA to protein. We're a step downstream.
Mm-hmm. There's a question getting into more the clinical trial. As you know, there's now a phase 3 trial for losmapimod. If someone is enrolled in that trial, will they be able to enroll in the FORTITUDE trial?
Right now, no. We have an exclusion criteria about you can't be on another investigational medication when you to come into this. And if you think about it, that trial is also blinded. And so we wouldn't even know if a person in that trial would be on losmapimod or placebo. And you can't really have two questions like that happening at the same time. And also because we both need to independently understand our safety and efficacy, you wouldn't want to have two investigational medications being studied at the same time. For anything that we see, we want to know, is that because of AOC 1020, not is it because of something else? That said, if losmapimod gets approved and it becomes a standard of care, then it's another question of, would there be a benefit of using these two medications together? Do you need both? Would one be better than the other? So down the road, I think it might be a slightly different situation. So if you think of other diseases like diabetes, most people take metformin, and when a new drug comes out for diabetes, it's studied in addition to metformin, right? So you would stay on your metformin at the same dose, and you would add the new thing on to see if it helps you in addition to metformin. So it is possible as more and more FSHD therapies come out that combinations or additional medications or things could be the way that FSHD is actually treated. We're not there yet. Let's get one. But down the road, you could imagine different combinations, and especially combinations of things with slightly different mechanisms of action, right? Maybe there's something that helps muscle regrow, and then we're there to block DUX4, and maybe that's the best combination. I mean, that's even more down the road, but there's a lot of possibilities you could think about like that.
Okay. Let's see. There are a bunch of questions here about tamoxifen. That always throws people for a loop.
Yeah, I saw that when I was talking. I was like, "Ooh, can I?" Yeah, let me try. So, tamoxifen and FSHD in humans, there's no connection that I'm aware of. That mouse model is just one very specifically that the way you turn on DUX4 is because they engineered a gene that it's like a tamoxifen part that turns on genes and then the DUX4. So basically, when you give tamoxifen, it turns on DUX4 in that mouse model. And that's just how we're able to manipulate how much DUX4 you get. I don't know of any connection at all in humans. So sorry if that was confusing.
Okay. And then let's see. There's some questions about if you're on certain medications, for example, opioids, because pain is a big issue in our patient community. Is that a exclusion factor for?
No, we want people who are on stable medications, and so whether that be for hypercholesterolemia or pain or things like that, as long as people are on stable regimens, that would be fine. I mean, opioids is a little bit, we just want to make sure there's no problems there. But yeah, if it's being used at a appropriate level, and that would be something that the physician who would be administering the study at the site would have to really try and understand. But there's no blanket exclusion for using opioids or other medications.
And I think an add-on to that, June, is also people are asking if they can continue their physical therapy as well.
Yes, we would. What we would ideally, like in a perfect world, what you would want is everybody to stay on exactly the same physical therapy regimen, exactly the same other drug regimen, exactly the same diet, exactly the same sleep. And the only thing that would be new is you add on AOC 1020, right? Because we want to specifically look at, if nothing else changes, this is the only new thing. How does that affect your FSHD symptoms? We know the real world's a little more complicated than that. We ask people to try to maintain kind of their same basic lifestyle, but recognizing, life happens. But for sure, if somebody is on a physical therapy regimen or works with a trainer regularly, ideally we would want people to maintain that as much as possible.
There was someone asking for clarification. How many muscle biopsies would be needed for this?
There would be two.
Okay.
Two. And for people who haven't had them, it's like there's a local anesthetic, a small incision would be made through the skin, and then it would be 4 pokes of something, and the needle is about the size of like a slightly thick spaghetti, maybe. So you get 4 little, not that long, like short little pieces of muscle tissue would be generally the way we would recommend that the sites do it. So it's 2 events of muscle biopsy that would happen.
Okay.
All
right, great. Thank you. There's a question.
You don't need stitches. You need like some Steri-strips and maybe a Band-Aid, and don't get it super wet for a couple of days. So it's not like stitches and a big surgery.
Okay. All right. Were there any off-target effects in the muscle, in the mouse model? And I guess connected to that is, if there were, are there any side effects anticipated or expected?
Yeah, I mean,
there's
always right. You always have to be really careful moving into humans. But what you do, and before you move into humans, is you do chronic studies in 2 species. So we've done studies in both mice as well as monkeys to just look for safety things. And up to the highest doses we studied, which were quite a bit higher than what we'll be studying in humans, we've not seen what they call any dose-limiting toxicities. So basically, everything looked really clean in those 2 animal models. So that gives us a lot of hope that we won't be seeing any off-target effects. You still have to do the studies in people, and we can't overpromise, but things looked really promising. One nice thing about oligotherapies is that they're very specific. Like the string of RNA that we use, there's not other things it can bind to just because of the way that the binding specificity happens. So it's nice that it is very much focused on DUX4.
Nice. There's a question. Can you explain what the booster is?
Oh, the booster. It's just a dose to try and get people to steady state a little bit sooner. So we think every 3 months would be what we would need to do long-term going forward, but just at the start of therapy, to maybe get people to steady state levels in their muscle a little bit sooner. So it's the same dose, same regimen. We just have 3 doses, kind of a shorter time frame before we go to a more spread out.
Okay. Question about someone has a metal plate and screws in their ankle. Can she still get an MRI if it's not of that part of her body? I don't
not
know.
An MRI technician. I don't think so. I don't know how far the metal has to be from the magnet before they would do an MRI, but basically any contraindication to MRI would be a contraindication for this study.
Okay. They don't even let you bring any metal into the room, much less anywhere near the machine.
Much less in your ankle.
Yeah. Okay.
But actually, let me clarify one thing. That's only because of the MRI. There's nothing about having metal in your body that would make the drug change, right? It's just for being in this study where we have MRIs, we need people who can do the MRIs. But down the road, if we get the drug approved, or perhaps even in another study that everybody didn't have to have an MRI, that person might still be a candidate.
Okay. Let's see. There's a question. There's a number of questions, not surprisingly, asking kind of part of it is, will this drug be available to people who are older than the age limit for the trial once it's approved? And related to that, what's your ideal, if all goes well, what's the time frame in which you hope to bring this to the FDA for market approval?
You do a clinical trial, what you're trying to do is minimize variability as much as you can. And so that's part of why you try and, I might have frozen again.
You're okay.
Okay.
I can see you.
Yeah, okay. You don't want to, you have to have somewhat, you're trying to minimize variability so that you can see the effects of the drug and not just tons of variability. And part of that is keeping it to sort of adults and not moving into much older populations for a clinical trial. And also, it is known that as people get older and older, they tend to lose strength just from aging. And so it's a harder population to study. And so especially in this first study, when we have a relatively small sample size, we want to try and keep the group somewhat more homogeneous just for the trial, just to help us get good answers about the study. But presumably once the drug would be approved, it would be for the treatment of FSHD. Knock on wood, we hope. And we wouldn't anticipate any sort of upper age limit. So that really is just a function of trying to do that first clinical study in a relatively small patient population, that we just need to keep it kind of a more homogeneous population in terms of age. And then the other question was about timing. I mean, this first study will be treating people for a year. And, because of the way we have the group somewhat staggered, because we want to make sure that it's safe before we move to the next group and next group, it'll take more than a year, probably more like a year and a half to 2 years to even treat everybody in the trial. We don't have to wait for everybody to get to the end, but we have some staggering. And then we would presumably need to do another trial after this, which would be another probably same amount of time. So it is a few years out. So it's hard to speculate on a specific date, but it's going to be, probably 4-ish years at the minimum. And that's if everything went very quickly. And yes, FSHD2 would also work for this. I just saw that question pop up, and that's an easy answer. People with FSHD2 would be eligible for this trial, and we will be providing genetic testing. So if people don't have genetic testing and they're interested in the trial, that's something we would provide as part of being in the trial.
Okay. The question, if a patient is using an ankle foot orthoses, would they be eligible?
Yes. As long as they can take it off and get an MRI.
Right.
Yeah.
Okay. And a number of questions around, is this therapy designed or intended to completely cure the disease, or just to stop and slow it down?
That is the big question, right? We don't know. We're in unknown territory here. What we feel comfortable saying is that, if you could theoretically stop any new DUX4 from being expressed in muscle, we don't think you would keep making those toxic effects. And so we don't think it should get worse, right? We think at the minimum, we would hope you would just kind of status quo, hold where you are and maintain what you currently have. But the question that really isn't answerable until we start doing these experiments, does the muscle have some ability to regenerate and repair itself if it isn't getting more sort of insults from new DUX4 coming out? And that just isn't known. So when we make our planning about how big of an effect we think the drug might have or how long we think a study should take, we plan that it would just stop progression. But all of us hope, and hope isn't really a very scientific word, but there is the possibility that you could see some repair, especially maybe in muscles that are kind of on the edge, that still have some regenerative properties, still have muscle stem cells in there. We just need to do some of these studies and really learn. And it's not just us, it's studies with other compounds as well that maybe have some of that potential. So I think over the next 5 years or so, we'll learn a lot more about all of that once we have these tools.
There's a question regarding if there's a limit on the BMI, body mass index.
Yes, the upper limit is 35. And again, that's based on the MRI machines, that many of them have that. Because we're doing these whole body MRIs, that is the limitation that we have in the trial, in this first trial.
Okay. There's a question about when you think the trial will start, and I'm going to add my own question. Is it now posted on clinicaltrials.gov so people can?
It is not. It's not quite yet posted.
Okay.
But over the next, very soon, which would be, I would say, we should get our first patients in the next quarter. We would expect to start enrolling patients at multiple sites in the US to start.
Okay.
Stay tuned.
All right. Let's see, we're getting close. There's so many scientific questions. It's interesting. We have a lot of scientists or science.
Let's do this again. This is fun.
It was fun. But let me go to one that's more of on the clinical trial end of things. People are asking about the open label extension. And it will, let's see, they're wondering about the dosing, because I guess you have graduated dosing in the trial. Like, what dose would someone receive in the open label?
Yeah. We probably would keep them on the same dose that they were on in the parent, but that's with some flexibility to change it if we learn over time that another dose is better. But to start, we would probably keep them on the same dose that they were on in the FORTITUDE trial. But you could imagine that extension trial lasting a few years, something like that. We don't know for sure, but it could go on for a while. And so if during the conduct of that trial we learn, oh my gosh, this is the best dose, not that other one, we would write the protocol in a way that we could get people to what we think is the optimal dose in terms of both safety, tolerability, and then efficacy as well.
Okay. And one last question about the open label extension. So people will be enrolling at different time periods and finishing the trial at different time points. So when someone finishes the entire trial, do they immediately go into open label, or does everything stop? And is there a gap before you have a start date where everybody starts open label?
No, ideally, yeah. I mean, in a perfect world, again, this would be like you would have your day 365 for the FORTITUDE trial. Your site already has the open label extension protocol and everything all set up. And maybe that day or the next day, you get your first dose for the open label extension. So that's how we're trying to set it up. We do have some provisions built in because sometimes there are delays with things that happen at a site, maybe getting them up and running, or things happen. So we will build in so that there can be a window for people to come in. But yeah, ideally, we would love to just keep everybody going. We think that would be the best from the safety and tolerability and also potential efficacy. If we are really knocking DUX4 down, we want to keep it down. We don't want it to come up and get it knocked down again, come up and get it knocked down. We want to keep it down. We don't like DUX4, keep it down, stay on drug. And that would be the ideal situation.
Actually, a few more quick questions. Is the trial eligibility just for FSHD type 1, or are you also looking at
FSHD 1 and
type
2? I see 3 or 4. I don't even
3 or 4.
I'm not sure I know about 3 or 4. FSHD 1 and 2.
Yeah.
I have some Googling to do myself here. Yeah.
Okay. Well, 3 or 4 would be, I think, probably defined by low methylation, but not having the SMCHD1 mutation, the other standard markers.
Okay.
So that might be.
Yeah, for now it's FSHD 1 and 2.
Okay. All right. And this one I wanted to include. Do you accept trial participants who have had cancer and are in remission within the last 5 years?
I'd have to look at the way the wording. It depends on the cancer. It depends on, we want people who are in a healthy state. So it would depend, like some skin cancers where you have like a surgery and it's cured and it's gone would be different than something that has a higher risk of recurring. And usually we do use a 5-year window to try and, before we say somebody would be eligible for a trial, but that would be something that would have to be discussed between probably the investigator and the medical monitor about the specifics. I can't comment on any specific kind of cases.
Okay. Great. Oh, wow. Well, we could go on for another hour and a half.
Let's do it again. This is fun.
And we will have to do it again, but I think I should wrap up. You've been so generous with your time, and I'll now turn it over to Beth to have some final words.
So we literally have hundreds of questions still in the Q&A. I'm wondering if we at least pulled them down from Zoom. Maybe we could at least address some of the other ones in a follow-up blog post or something like that. So we'll attempt to get to everyone's questions. We will definitely do that. But I do want to thank
email Alyssa. Show that email again.
No, there's hundreds.
Actually,
Marissa.
Don't email her individual scientific questions. That would probably kill her if she got 100 more questions. But know that we do really want to keep this dialogue going. As you can tell, we're super excited, and it's great to see this engagement from the community as well. So, really appreciate it.
Great. We have a very amazing community. I love our community. And I want to thank the community for attending our webinar today. Very, very special thank you to Amy and Alyssa for these really exciting updates. We're all just chomping at the bit for all of this coming down the pipe. And thank you so much for answering so many of our questions. I appreciate it very, very much. Just real quick, peeps, our next FSHD University webinar is actually next week on Thursday, the 23rd. Our guest speaker is Dr. Rita Perlinguero, I love to say her name, from the University of Minnesota. She's going to talk to us about stem cell research. So you definitely don't want to miss that one either, because all of it kind of ties in together. Just be sure to visit our website events calendar for all of the upcoming chapter, wellness, all these educational events. We have so many. Again, thank you everybody for joining us today, and we will see you next time. All right. Bye-bye.
Bye. Thank you.
Thanks, you guys.